Right, so really delighted to have this presentation. I'm going to introduce Dr. Hanley and she'll introduce the rest of the panelists tonight. But one of the things that we do is we do clinical research, and this is what this is about. And it's really important to have all the resources be at your fingertips. And so we're really important in the presentation. So Dr. Hanley is professor of neurology, neurosurgery, anesthesiology, and clinical medicine at Johns Hopkins University. He is professor in the School of Nursing as well. Director of the BIOS Clinical Trials Coordinating Center, which is an academic research organization now 25 years old, as you understand. And they've designed and developed and conduct many clinical trials. BIOS is embedded in the neurology department, but it actually works through other departments and has clinical trial experience beyond the field of neurology. And as we'll hear about tonight, even in obstetrics and gynecology. And this is beyond Dr. Hanley's interests, which are primarily neurology. So the seminar series, GORS, which we've been doing for about 10 years, we haven't had this represented, but I think it's really high time that we did. And with all the changes with the new administration, it's increasingly important that we optimize opportunities and expedite what's going on. So I'm looking forward to it. So thank you very much for coming.
Thank you. So I think it was a very nice introduction. I was lucky enough to meet Jenna and her co-PI Meredith as RAP started. They were a triumvirate with Eric Jellin, who died, and they took over. And how is a neurologist allowed in the important area of maternal fetal medicine and pediatrics? And that comes about because we did on our own with the group we're going to talk to you about tonight, 10 or 15 trials in the neurology area. And we learned the lessons of all the mistakes you can make without having help on hand. So we don't take over anybody's content and specialty knowledge. We help people project it in a turnkey kind of way. And what I hope you take away, we're going to talk about some of the complexity of clinical trials, but I hope what you take away is that there are solutions to the problems that you run into as you're attempting to ask one big question that could potentially gain knowledge that would change practice. So that's sort of the story tonight. And
here, you click that one. I'll click this
you
one.
click that one. Okay.
You click.
All right. Oh, I clicked down here. Got it. Okay. So the people who are going to talk, Jen is going to represent the PI group, and she's the maternal fetal medicine, fetal and pediatric surgeon, and then pediatrician. So there was a triumvirate of people bringing their expertise. Nolene, I'm Nolene now, unfortunately. Nolene was coming down from Philadelphia, and she was the manager for the trial, but Amtrak has stopped because there was an accident. So I will give her presentation. And Jessica will tell you at the end a little bit about if you want help, how you can get it with your idea or a group of people's ideas. And then we'll have a panel in which Nicole on the far end and Lindsay, who are also faculty members, but they're also people who are trial scientists and who can help solve those problems we talked about. They're going to be able to give a little bit more color to your questions. And Meredith can't be with us tonight. I got it. Okay. So I said we did a bunch of trials. On the left are some of the things that you need for a trial. If you look on this tree, each major branch has a theme. This branch here is one around elevated intracranial pressure and hydrocephalus. And pardon? Can you see that or not?
They don't see your.
Oh, they can't see the
I
pointer.
was gonna try and mirror it.
Yeah, good. And this trial here, HENS, just finished this year and just got presented worldwide last week and just came out in the New England Journal. The other little white ovals are other trials that have changed practice. So we've been in the business for over 30 years, and we're now sharing our expertise. We really do three things. We do trial planning, and you're going to hear a little bit about that tonight. We do grant organization after we have a plan, and that provides the specificity to a plan. And then we do trial operations, running the clinical coordinating and or the data coordinating center. And we do feedback. The things we learn about what was, to use a technical term, crappy about the plan, written plan, we then fix the next time we write the plan. And we found that taking that same continuous quality improvement strategy that we use in medicine works in trials too. And I've already introduced people, so I think we're going to move to our first speaker, which is Jenna.
Okay.
And let's switch.
All right, great. So it's great to be here today. I did this course when I was a new faculty member as well, and really it's one of probably the most valuable things that I've done here at Hopkins as a junior faculty member. So thanks for coming today, and I hope it's the same for you. That one was mine right here. So yeah, thank you. So I'm going to share with you today a bit about our RAF trial consortium and what we've learned through doing the RAF trial so far. And I'll share with you the first results that we've published as well. And I'll acknowledge Meredith Atkinson, who is the co-PI currently, and she's a pediatric nephrologist. And so I can't imagine doing this without her. I mean, she is like my Siamese twin when it comes to this. We have been through it all together. And so everybody here is going to be familiar with the most severe form of fetal renal failure, which is bilateral renal agenesis. And when we were thinking about targeting this condition, we really needed to deal with some of the most severe of all of these cases when we were thinking about this intervention that I'm going to share with you. And how frequent is that? Well, it's actually pretty common overall. And so when we look at all of the births in the US, about 800 cases of renal agenesis occur per year. And when, let me go back one. That skipped a lot. But there are many other causes of early anhydramnios during pregnancy, so that are a result of fetal renal failure from other conditions. So whether that's urinary tract obstruction, multicystic kidneys, dysplastic kidneys, all of these are much greater contributors to this problem. And so we needed to study this group together as a whole. And one thing that's really challenging about fetal renal disease is that in a way, we think about it, it's a double lethal disease, right? Because the kidneys create the anhydramnios. So we have a child that has renal failure, but during the pregnancy, the consequence of that is pulmonary hypoplasia, right? From the compression. And so the immediate lethality comes from the pulmonary hypoplasia and the inability proper lung development through the pregnancy. So that makes this condition, when we target it, doubly complex. And so the question that we're really addressing in the RAFT trial is, do amnio infusions that are performed throughout the pregnancy prevent lethal pulmonary hypoplasia? That's the question of the RAFT trial. There's a lot we would like to know from the RAFT trial, but this is the primary question. And when we think about how this developed, well, it started, there was a very famous case report many, many years ago that came out of Hopkins, where a congresswoman's pregnancy was treated with a few amnio infusions. That baby was a survivor initially, went to the NICU, ultimately has a kidney transplant. So this created a whole lot of enthusiasm and excitement in the lay press. We had a lot of calls for this to be performed clinically, and we had to really think about how we were going to address this as a group, as an institution, and as a scientific community. And so what was available at that time was really just these few case reports, one here from Johns Hopkins, and a few other series of the potential that this could be a survivable condition. And so around that same time, there was also a research workshop that was held by the NICHD in combination with NIDDK that deals with kidney disease. And they proposed a research agenda, which we covered a lot of topics surrounding this problem of pregnancy, early pregnancy and hydramnios. And so a pretty clear research agenda was established from that. Next step is we had an ethics summit here, because as you can imagine, this creates a lot of, people have a lot of feelings about this intervention. And so we held a research summit with all of the important stakeholders. So the doctors, the nurses, the ethicists, the insurance providers, all of the people who would intersect with these patients to think about what was important in trial development. And so all of those kind of parts and pieces was the foundation for really creating and the framework for the RAFT trial. And also Eric Jellin, who was really like the leader at that time, who kind of brought us all along, no matter how much we were kicking and screaming about it. He really was the force behind getting this together and interacting with the group that you see here and forming that relationship. And so how the RAFT trial developed is it's a non-randomized, open-label, multi-center trial. We started with 9 sites. We ended with 13 across our consortium of fetal therapy centers. And we performed serial percutaneous amnio infusions to study if that actually improves our initial survival. And so we had 2 arms, one for renal agenesis, one for other causes of fetal renal failure, and an expectant management group that was only to document the natural history of the disease. So it's not a comparison. We know these babies die, right? So there's no proof of concept that's needed with the expectant management group. But they're not really well characterized in the literature besides what we're familiar with telling our patients. And so our primary outcome was neonatal survival to 14 days and placement of dialysis access. And so this is really just a surrogate to tell us that we were able to avoid pulmonary hypoplasia and that these babies were able to receive their initial treatment for fetal renal failure. And so that is the primary question. Do we avoid pulmonary hypoplasia? And we also know that doing a single amnio infusion is pretty safe. It's 1 needle, local anesthesia, pretty straightforward. But what we didn't know was, what does that safety profile look for pregnant women when that's done multiple times during the pregnancy? And so that is really our #1 aim of this trial: maternal safety and feasibility. Could we do this multiple times during the pregnancy, and what happens obstetrically? And so we didn't know that. And so this is the main focus. This is a maternal safety trial. And then secondarily, we wanted to know what happened to the babies. So how effective was this strategy of overcoming lethal pulmonary hypoplasia? And #3, long-term, do these babies get to transplant? Because that's their ultimate, that's the ultimate cure, right? Or the next bridge. And then finally, the few other things that were included: how many got to discharge, how many were on dialysis, and to get to that final time point. And so what this trial looked like, it was done under a central IRB here at Johns Hopkins. And we had a lot of patients that would reach out. We did a pre-screening assessment to see if they met our general inclusion criteria. And then if so, then we invited them for a screening visit at a RAP site. They had to undergo all of the consultations to understand what caring for a child with renal disease would look like. And so they met with, of course, MFM, the whole pediatric teams, our genetic counselors, also our social workers, because this is a major impact for families when they're considering this intervention. They had to start their enrollment by 26 weeks, and we had to begin amnio infusions. That was actually a pretty big barrier to getting patients in the trial, because you had to make the diagnosis, get through your genetics, have all your consults, make sure the baby looked normal, and then begin this infusion. And then we did serial amnio infusions to maintain normal fluid as much as possible up until delivery. They had to deliver at a RAP site. We measured our primary outcome there at 14 days with other survival time points of 30, 60, 90, and hospital discharge. When patients were discharged from the hospital and on chronic dialysis, they received quality of life study surveys, because we really want to know what the impact was on these parents and on these families and the children, right? Now, one thing that was also a gap is it would be really nice to know what the quality of life and what their impression was from that time period, from birth until their discharge home. And more to come on that. And then finally, the last measure is survival to transplant. And currently, out of our whole cohort, we have 8 transplants that have been performed so far, 5 for renal failure and 3 in our renal genesis group. We intended to enroll 70 maternal fetal pairs, 35 with bilateral renal agenesis and 35 with fetal renal failure. What was also unique in this trial is in July of 2022, the DSMB asked us to pause enrollment in our BRA arm. And that's because we did have some questions. We were seeing a pretty good result from survival, but we knew that these kids were going to be medically complex. That was not a surprise. To start with, all the babies are dead. So we expect them to be complex, but there was the concern on the morbidity and mortality in this group, and we had demonstrated our primary outcome. So they asked us to close that, or to pause that arm, and as an investigator group, we decided to close it and try and get the results out to the public domain as quickly as possible. So that happened after 21 enrollments, 18 in the intervention, and 3 in expectant management. And so this is what at the end, now our study is closed. It is finished. We are in the data cleaning and analysis stage. And so through the whole course of the trial, we assessed 523 patients. We had 189 come to an on-site, to a site for screening. And out of the enrolled patients, we ended up with 21 in the renal agenesis group and 34 in FRF. And not surprisingly, those that did not enroll most commonly were for other anomalies. We couldn't start by 26 weeks, or they had pregnancy termination. And so this is what we've learned so far, is that it was actually really successful to get to that primary outcome. So we had 17 of 18 born alive, and 82% of them survived to the primary outcome. And so that clearly was much higher than the 30% we anticipated in our power calculation. And not surprisingly, the obstetric complications that we observed were some bleeding, membrane separation, and about 60% or so had PPROM, which was a big trigger for delivery. Other things, that the time from amnio infusion starting until PPROM was obviously much longer in those that survived versus those that didn't survive. And so that's the combination of the patients that are survivors are in dark blue, and those that were non-survivors are in gray, just to orient you to the results. And so if you had a longer time of amniotic fluid, you were more likely to survive. And we didn't observe any major safety concerns. In addition, we also learned that the more amnio infusions you had goes right along with that. The longer we can maintain amniotic fluid, the more likely you were to survive. And it happened a lot. Like the median number was 14 in our survivors. Our large, and our range is still pretty wide, 11 to 18, and we had a few patients that have more than 20. And these children were born later and had a higher birth weight, which also makes sense because they need to then be cared for with peritoneal dialysis. But one of the big things that we have learned and that we're really working on is that it allows us to characterize a whole new novel cohort of infants. And so this was a group of kids that we never have taken care of before. There are no rules about how to manage them clinically. And so the second part that is really driven a lot of the work following our initial question is what happens to these children and how should we care for them for the future? And so this table you can't read in detail, but it does describe what happened to those babies that didn't make it and what complications they faced. 6 of them out of 17 survived a hospital discharge, which was a long time, on the median time, 24 weeks. 2 died after they were discharged home. And out of the 4 that were remaining, I'm sorry, I had an update to the slide that I guess I didn't do quick enough. But 3 of them also had strokes. And so that was one of the big mortalities or morbidities that has caused a lot of excitement and a lot of enthusiasm from this time. And that's something that we've worked a lot on to understand better. And out of the 4 long-term survivors, 3 of them are transplanted. And as I alluded to, stroke was a big concern in this group. And so we wanted to understand this a little bit better. And so for us as a group here at Hopkins, we had to look in our own cohort, because part of what we didn't have in our primary trial question was clear characterization of the postnatal course of these kids. And so, the things that would have been nice, right, to have their imaging. And so it allows us to we added being able to upload postnatal imaging, and so that we could capture some of these things for those that were longer term survivors. But in our own cohort here, we had a number of stroke episodes that were observed. And these children obviously have a lot of risk factors for that. They have significant hemodynamic instability, clear endothelial dysfunction. And so many physiologic reasons put them at risk for stroke, and there's no comparative data. We actually don't know kids with renal failure from birth what their incidence of stroke is anyway to compare to. So it's really in uncharted territory. And so one of our neurologists here has done a lot of work and joined our team, helping to understand that a little bit better. And so it seems to be that these strokes are not always recognized clinically. Sometimes they show up as seizure. And when we do that imaging, we find that they might have had a stroke episode previously. And so it's much more common than really clinically recognized. So we recognize there's still a lot of work to be done. And that really brings up the question of needing to follow these kids long term, and what does this mean for their neurodevelopment? And so there's more to come, and that's really the big driver of the next clinical question for us. And as I alluded to, we didn't know how to take care of these babies. And so across our trial consortium, our neonatology group has been phenomenal. They are so engaged, and each of the subspecialists has their own work group that gets together to talk about cases, to help work through how should we handle a lot of clinical scenarios. And so that has been a huge secondary gain as a result of this trial as well. And so within our neonatal work group, they did a survey to, just catalog, how did they take care of these kids? What did they do? So because none of the neonatal management is protocolized within the RAP trial. And what it allowed us to do is try and start to figure out where are some spaces for harmonization so that we can work to develop some practice guidelines and maybe some best practices. Now, I mentioned briefly the quality of life surveys. And so what we know from our parents so far that complete those surveys is it's similar to what we expect from families that have medically complex kids. Everything takes longer. parents are stressed, they worry about the future for their kids, and they have a high level of anxiety. But since we have that gap from the time of hospitalization until discharge, in order to understand that better, we also have started a quantitative study to interview these patients and really get their reflection on what this process has been to them. Because as clinicians and providers, we have a lot of assumptions about what it means to be in these parents, roles. And so we need to listen to what they have to say also. And so we are interviewing all of the patients that were included in RAFT, no matter what happened to them and their outcome. And it covers all of these features. So how they felt that they were prepared, their counseling, their consent process. And so far we have 5 of those completed. But in order, now that RAFT 1 is done, there has been a lot of enthusiasm in the community for this intervention, and it is being offered in a lot of places that are even outside our RAFT trial sites. And so as a community, we also had to think about how do we deal with that? Because now we have a lot of questions about the kids and what the neonatal and what the pediatric consequences are. So, we're in this space now of the RAFT registry. So in order for us to continue to collect data and observe the outcomes, we're doing this under a registry umbrella with some similarly, some of the same milestones and endpoints through our North American Fetal Therapy Network. And we're hopeful that that will continue to inform our future clinical management. It's much simpler of a design because it is a registry and is aimed to collect data at specific time points. And that's where it comes to RAFT 2. So we are working on the development of that trial. And so I'll share with the team here that is instrumental in helping guide us to develop that protocol successfully. And it takes a whole community to run a big trial across centers. And so by all means, we have a huge support of our research staff, our PIs, our coordinators, our other subspecialists. And we always acknowledge Dr. Eric Jellin, who really was, as I mentioned, the force that brought us where we are today. And finally, just a little bit about some of our kids. And so our parents love to share their stories, love to share their pictures. It's all of these kids in these pictures are transplanted and developing well. So along the way. All right. Next.
So there's a lot of accomplishment here, and the one thing that Jenna, I think, was too modest about is this is in the process of actually changing practice. And practice change is complicated. Particularly in maternal fetal medicine, where you got 2 patients, and the first signal is this appears to be safe for the mother, and can be helpful for some of the patients. But question is how many deaths are there going to be along the way? But the world in terms of parents, and many, but not all physicians, are responding to the JAMA paper, and there's an increase in use now of amniotic infusions. And like in everything in medicine, there are physicians and inventors out there trying to make devices that do this better, and there are people who have solutions to some of the problems that have come up. And there are people who say maybe we shouldn't be doing this at all. And it helps to have the community that was in the next to last slide to get all those questions asked, so that when you make the proposal for the next trial, you have at least legitimate answers, and you take the position that if we don't do the trial, we won't have the data to answer your question. So Nolene was going to talk about doing the trial, and we all learned things from doing the trial. Nolene is a project manager who's been with BIOS for about 20 years. And she worked from the beginning on this trial. So I'm sorry that she can't give the presentation. And you heard the story, but there was a trial consult, there was planning and development, and then she's going to talk in this talk that I'm going to give more about implementation and getting to an end and having a single result, which is this experiment's no different than something you do in a week in a cell culture dish. You get one question and one statistical test for that question. There have been lots of publications, and those are all hypothesis generating for the next trial. So we're going to talk about a little bit about publications that close out. And here's that three-step set of tasks that we can help you with. And currently, we're helping groups of investigators that have multi-center clinical trials through an award from the National Center for Advancing Translational Science, NCATS. And we're part of a national group that's called the Trial Innovation Network. So currently, everything that was able to be done for RAFT was free until they got the award, and then the award paid for the trial. So you can see there's multiple steps in trial planning and in developing a research strategy. And we're going to hear a little bit about the consult that's going on for RAFT-2. And the big issue there is matching the data we have, both the primary and the secondaries that we've collected, to a good hypothesis that's interesting to the Institute of Child Health. So Nolene said, where does she start in telling you the story? I think she wanted to say that NAFNet was a wonderful group to work with. They had come together recently over the 10 years prior to RAFT, but they hadn't done an investigator-sponsored trial. They'd done some commercial trials.
A number of things. So there was kind of a concurrent investigator trial, but it was probably along the same timeline.
Yeah. So this was new and was embraced by NAFNet.
And also what was unique within NAFNet is everybody, all the centers agreed not to do this outside of the trial. And so that was another really big thing that diverted patients into the trial center.
Right. And after the trial occurred and practice changed, it predicated the need for registry. So we used the site assessment instrument, the SASE, to assess for Eric and Gina and Meredith what the capabilities were at each site. And we assessed three domains of capabilities. The study PI's capabilities, the study teams, and how the coordinating center would interact with people at each of the sites, because you have to contract with them and you have to get approvals at regulatory approvals at the site. And the SASE instrument helps what for most people is a disorganized process, and gives the investigators leading the consortium a list of potential partners and the pros and cons of each one. And we can turn that into a numerical score, but there's also qualitative information there. The second big thing that there we go, that we did after we got the sites on board and they'd been chosen by the PIs, is we help them start the trial. And most grants are for 5 years. And I think it's not simple math. If you take 2 years before you ever get to enrolling your first patient, you don't have a lot of time left. And it's very hard to get that coordination from our university to 20 other universities. In this case, I think it started out with about 12. So we've written some rules of engagement, starting with that information about who do we actually talk to get a decision from the other universities. And then what do all of the researchers at the other sites need to know about the RAF trial in order to start their first patient. And we've been working with that for the last 8 years. It's called Accelerated Startup or Accelerated Startup Unit, ASU. And our goal, which we've reached over a series of trials, is to be able to get most of the sites to start in 90 days, which gives you, let's just say the industry standard is somewhere between 3/4 of a year and a year. It gives you another 15 or 20% of the time that your money is paying for to do the actual work you want to do. And it works in 3 domains. The IRB domain, and if you're at Stanford, you can sign on to the study, but you're still going to look at what's the risk to Stanford. So the sites have to evaluate that, even though there's a central IRB. Contracting, they're going to be people looking at the same thing. Are we getting paid enough? And then there's a whole set of regulatory things, most of which are around good clinical practice, so-called GCP, where that's really the training of the people on the sites to do the work. So the SASE addresses all 3 of those. Now, these were the original sites, and I think there are 12, is it?
Started with 9.
9?
Ended with 13 because early there was nowhere to go in the middle.
Right.
And so trying to add some minutes.
Right. And these are some of the domains of how we helped both the PIs and the sites. So you have a team at BIOS that helps with the protocol amendments, changes in research, and annual continuing renewals, which you have to do with your IRB. A group that helps with site training and the onboarding of the site with all of those regulatory accomplishments. And then we monitored, because this was a safety trial, how the mothers were doing, what events they had, and that created the PPROM and bleeding data that you saw. We had to monitor in real time, so the fourth column over is biweekly reporting of knowing where you stand, so that if you go into the lab and you do an experiment, you don't look at the results, sort of shame on you. And the same is true here. We can't let major maternal or fetal injury go unidentified. And then we have to report that to an external group, the Data Safety Monitoring Board. So organizing that for the PIs is important too. Early on, this set of green dots is the enrollment. Things slowed down, and so the PIs made the decision to add some sites. And we reused that SASE and we reused that onboarding a second time to bring on another 4 sites. And that way we ended up with 13.
13.
And those are those sites. We now have enrolled the last patient in February, I think. So the data has to be monitored, and all the missing points either have to be, people have to search for them or say they don't have them. And where there's uncertainty in what the site said, we have to clean the data and eliminate the uncertainty. And at the same time, we take the data safety monitoring report and we turn it into the statistical analysis report for the primary and the secondary outcomes. And the primary outcome was survival at 14 days and ability to have a catheter to start dialysis. And that's what will be reported in the second group and in the total group in the next publication. And it looks like what happened in the first group, except survival's a little higher, which may just have to do with the learning of taking care of these complex kids. It may have to do with the difference between fetal renal failure and agenesis of the kidneys. So after all the data is in from the sites, you can close the sites, and then we begin to do the analysis. The secondary analyses will be part of the paper, and a lot of people will grab onto them as things that should go into guidelines. But you know, one thing you should take away from this particular session is they're secondaries. They weren't a priori hypotheses. So even though the people who did the trial are more experienced, their experience is limited to 55 patients now. So the secondaries are what they are. Gina said it was a good experience. So the best measure of it is that they came back and wanted help with the next one. And Jessica's going to tell you.
I think I can do both. We'll see. Like having my head and rubbing my stomach at the same time.
Well, let's see you do that.
Okay, I'll give it a shot.
You want me to?
It's okay. I'm gonna.
Lindsay will do it. She doesn't trust me, but she trusts Lindsay.
Yes.
And that tells you
I've been working with you
something,
now for 4 years. so-
something very important about the organization, okay? The people who run BIOS are very skilled.
We won't tell them what our nicknames are yet, maybe
No.
at the end. All right, hi, everyone. I'm a consult navigator with BIOS, which I swear is not a made-up role. I'm going to tell you a little bit about that and what I do with our investigator teams. And really what I'm going to tell you about is kind of where we go from here. So you heard about the science of RAFT1, and you've heard about the innovations and operational innovations that we use to enhance how we manage RAFT1. Where do we go next? So RAFT1 laid the groundwork by demonstrating the safety, feasibility, and initial efficacy of the intervention, generating a robust data set to inform future hypotheses and research directions. This opened the door for RAFT2, where the goal is to explore long-term outcomes, assess real-world effectiveness, and generate translational data that enhances generalizability. So to bridge that gap between what we learned in RAFT1 and the questions that we have to answer in RAFT2, the investigators are currently participating in a consultation with the Trial Innovation Network. And I'm going to speak more about what this entails, what the consultation is in a moment. But here, I just want to emphasize that the overarching goals of the consultation are to demonstrate community equipoise and to develop a trial that focuses on transplant efficacy, both of which are supported by data generated from RAF1. So before I dive into the details of our consultation process, I'd like to orient you to who the Trial Innovation Network is. So the TIN, like Dan already mentioned, is supported by the NIH, specifically the National Center for Advancing Translational Sciences, and it's a collaborative network of 65 CTSA hubs and 3 innovation centers. Hopkins is actually both of those things. So we are a CTSA hub, which is the Institute for Clinical and Translational Research, or the ICTR. You may have heard of that. And then we're also a Trial Innovation Center, which is housed within the BIOS Clinical Trials Coordinating Center. So BIOS is a coordinating center and also the JHU Trial Innovation Center. And in our role as the JHU TIC, Trial Innovation Center, we provide consultations to investigators developing their multi-site clinical trials. So this figure here, at least I like to refer to as our washing machine figure, and it illustrates our TIN consultation process. So the investigators come to us with their dirty laundry, which is the early stages of their trial protocol in the early stages of development. It goes into the consultation washing machine. And during the consultation, the consult team, which consists of both trial operations and subject matter experts, provides recommendations aimed at strengthening their study design and ultimately produces a nice, clean, robust clinical trial. Now, there's 2 different levels of consultation that you can participate in. All investigators first participate in an initial consultation, which typically includes 2 to 5 one-hour meetings. During these sessions, the consultation team provides tailored recommendations across the resource areas listed here. And I'm not going to go over specifics of each one of them. They can really be summarized by the final row, which can you even see? Yes, you can see it. It's highlighted in the blue there that for every initial consultation, every investigator receives individualized guidance to enhance and innovate their multi-center study design. And the word there that I really want to take a second to emphasize is individualized. It's not a one-size-fits-all kind of thing. You come to us, you tell us what you're looking to get help in, what your problems are, what issues you need to fix, and we customize the consultation to address what those needs are. The next level of consultation is the comprehensive consultation, which is currently what the RAFT investigators are engaged in. As this name suggests, this process is more in-depth than the initial consultation and is guided by the set of deliverables shown on the left side of the screen. These deliverables represent the foundational components of a trial protocol and ultimately support the development of a grant proposal, which is the end result of a comprehensive consultation. We're looking to submit a grant proposal. So these deliverables represent, we use these deliverables as a framework to structure and guide the consultation process. You can see here how these elements have been put into action for the RAF2 consult. So far, the TIN has supported the study team in developing the RAF registry, analyzing RAF1 data to generate hypotheses and inform the RAF2 protocol, conducting an ethics review, and facilitating communication with sponsors and stakeholders. And with that, I just want to say that we are open for business, and we're excited to support you as you develop your clinical trial. You can apply for a free TIN consultation. It is free, using the link provided on the left side of the screen, or you can reach out to me directly and I'd be happy to walk you through the process. If you're unsure whether a TIN consultation is the right fit for you, we recommend starting with the Trial Advisor Program, which is on the right side of your screen. I don't know why I think it's backwards from what I'm looking at on the screen. Which is available for all Hopkins investigators. And you can kind of think of the TAP consultation as the light version of the TIN consultation. So it still offers expert recommendations on your trial design, but it's typically through fewer meetings and a more simplified process. And in many cases, those TAP consultations serve as helpful starting points and often lead to a recommendation to apply to the TIN for further consultation.
And they're through the ICTR.
Yes.
And BIOS.
And BIOS.
Right.
And that's it. That's the end.
That's the end. So, we're open for for questions at this point in time.
From the audience? I have to say it's terrific. And I can understand, why you did it, because I think at that time in our department, I mean, I think that it was crickets probably.
Well, it's like, as a clinician, you don't have the training in like, this isn't what you know, and you don't know how to do it well, right? And so, I think we can execute a lot of things with good guidance, and so I think that's really a big part of it. I mean, all of the people at the TIN and BIOS, they are complete experts in their space and have tremendous experience with all these things. And so you can be motivated as a clinician, as an investigator, but it really jumpstarts you into something where you can be successful.
This goes beyond journal club, and it goes beyond.
This is it. So.
No, let me say one other thing. It goes
DCC basically, but
Yes.
it's a DCC on steroids, because some of the DCCs just do the DCC part.
Mhm.
And so,
and it goes beyond a statistical consult,
yes, of course.
and it goes beyond getting the very important help from people who are doing single site work.
Believe me, yeah. I mean, the people in the audience here that have done it the hard way.
including all of us up here.
Yeah.
And we're committing to try to make it less hard.
And our goal in the TIN is truly to not only help investigators like yourselves, clinician investigators, that's who we want to talk to. But it's also for us, the innovation part, our middle name is to find ways to improve clinical trial efficiency. How do we do them faster? How do we get them done on time, on budget, or even ahead of schedule to get those answers to the questions being asked and data supporting out to the bedside, to the community faster? What's the next question? Rinse and repeat. Let's keep pushing the bar forward. So that's where we focus as clinical trialists, but we also make connections. And we're also here to educate, and build capacity. So we like to hope that those, like our RAFT collaborators, have learned things along the way and continue to work with us through consultation, but at the same time, they've grown and they're able to be part of the conversation. So we're able to get to answers faster because the education has happened as well. So it's very comprehensive, multidisciplinary. Like Dan said, we take that approach from medical school and apply that here to the clinical environment as well, clinical research environment.
Wonderful presentation. Thank you. This is what I do day in and day out, by the way, and it's so much fun. I was able to reduce my onboarding time to 1.5 months. But 2021, when we were onboarding the first single IRB studies, it took maybe 6 months because nobody was aware of what to do, including our own IRB. And now, last year, we were able to onboard in 1.5 months on average, and some of them even earlier, which has been a lot of fun. I have a study-specific question for you. For the registry, are you also looking at the quality of life impact of people who do not want this intervention?
So that, that's a really hard space to capture, because we collected as much as we could about our screening group. Because a number of, if we pre-screened 500 and we had 189 come on site, that's a big gap. And we do have within the red cap questions about why they didn't come. And so some of it was a clear exclusion. Some of it was choice. Some of it was decide for termination. So we have very limited information about that. But
is there a way to then channel them to a different independent arm? Because if they don't want any intervention, which could be standard of care or the innovation part,
right.
Maybe there's a way to capture that information, because that is the external control,
Yes.
which will give you a very clean assessment of how the quality of life is being impacted by the intervention.
You're right. And so that was one of the things we talked about within the group developing their registry. There's like, do we have the people who we even screened for?
That's literally what,
Yeah.
the registry, you have the option to do that.
Yes.
That would be beautiful.
And it would be great if we had like some funding to do that. But it was one of the things that we had to make the hard choice to not have a separate pre-screened non-enrollment group. And so we're hopeful, we think that's a really good point, really thinking about planning for RAF2. Because capturing that group that doesn't enroll and understanding that is important.
It was part of RAFT 1,
Yeah.
and that's that very low line on the recruitment. And we ended up
Oh,
with
and as far
5.
as, yes, and we did the expected management group.
Yes.
Right.
We only had 5 people that agreed to that.
So that's a measure
Was-
of how difficult
Yeah.
it is.
So,
So
yeah,
another way to do what you're saying is important. Another way to do it is a separate, broader study where you start treating each individual in a similar way. And that would be a survey of people, both who underwent further treatments and who didn't.
Fantastic stuff.
yes. No, we expected a lot of more patients to enroll in expected management.
You are very optimistic.
But it was,
And what you point out is true of any trial, okay? The trial refines a population to the people who are participating. And we all have a right to not participate. And we don't even have to tell you why we didn't want to participate. So even if you do 3 or 4 trials in a row, you still don't know completely about those people who aren't participating. One of the advantages I showed you early on that tree of some of the many trials that came out of the early brain intensive care unit and then BIOS. One of the things that happens there is, and it's happened in RAFT, is people come back and ask the next question.
Yeah.
And that's very important because it looks at reproducibility. And reproducibility is going to probably be highlighted at NIH in the next 4 years at least, probably longer, because it's important. And the RAF group is struggling with, they want to do most things the same way they did in RAFT, but they want to and get a test of, is this reproducible again? But they also want to ask some new questions. And then the main new question is, how do we help these neonates get to the end goal of the amniotic infusions, which is to get a kidney transplant.
And just out of curiosity, I'm not a pediatrician, I'm a very lowly internal medicine doctor by training. So do they require another kidney transplant after a certain age? How do you
we expect that they're gonna need at least 2 transplants, because they will be eligible for transplant by the time they have to be about 20 pounds. And so nutrition is a big problem with these kids. And so our common transplant age is somewhere minimum 2.5, getting towards 3, some even later. And so we expect that kidney will last them, hopefully till adulthood. And then we expect they might need another transplant later. Cool, thank you. So what I learned is what's great about in pediatrics, their time on the transplant list starts from when they begin their dialysis. So once they're finally listed, they get that bump for that whole period of time that they were on dialysis. Okay.
Question?
No.
I have a question.
Okay.
No one else doesn't.
Thanks.
So actually, several, but is it primarily through NIH that your focus is at this point for the BIOS?
So
No,
Nicole's-
I mean, the question of sort of kind of with all the changes that are coming with the administration, I think maybe there's a different.
It's a little more challenging, yes. Yeah.
It is a target.
Mhm.
Yes, so BIOS hosts the 10. So is the mother organization. We at BIOS wrote that grant. But we also work with pharma. We have pharma trials we're doing. We work with device companies. We have device trials that we're doing. We've been involved in about 4 pharma approvals. We've been involved in 7 device approvals. And we bring that regulatory and commercial background as well to the issues.
I think, that's like a good enough. I think that would people maybe have interest in. Well, and then the way it's laid out, the 10 would happen sort of earlier in the ideal, that's the ideal. And then there's reality, which is that some grants get submitted without this kind of thing. And my question is that something that the investigators can kind of pop into, let's say, on a grant revision, and then get some expertise there, and then also get a BIOS sketch and stuff like that too, because I know individuals, not in our department actually, who are struggling with this kind of thing. People, Jim said, the scientists and the docs don't know necessarily what it is, but it's not necessarily an individual clinical trial, but it is a clinical study. And so I want to make sure that that would also be something you would.
You want to speak or do you want me to? Okay. So Jessica takes people who've had 1 strike. Okay.
Once, right? So that's kind of what I'm asking, but also not for like an intervention, but tissue observation.
So
We do.
we just, Jessica, last week just submitted a longitudinal study with gene proteomics and clinical phenotype. So yes, we do those. We like biomarker studies. And we are particularly good at first in human with drugs and getting studies to go up to phase 3.
Just to clarify a little bit, to add on to that, we're disease agnostic. So Dan used the terms earlier, we're clinical trialists. We're looking at the clinical trial science. So you individuals, clinician investigators bringing forward an intervention, a trial, or a study of some sort, or a biomarker or other, we're not one size fits all to where we can't talk about those things. We're going to talk about feasibility, budget, operations, workflows. And if there are clinical, biological questions that you need answered that we don't know, it's about those making connections again. We're going to tap into our consortiums and our Rolodexes to bring those people to the table so that you can then benefit from those. And you said, add a BIOS sketch. It was described earlier, the consultation is free. Once you get to a comprehensive consultation, which will end with a grant submission, that is a collaborative grant submission. So you're going to be writing that grant with us. We're going to be embedding into your trial and your grant application all of our infrastructure and workflows with a budget to do those things, but you're also then benefiting from the research experience for your reviewers to look at as well. So whether that's before your first application, you've submitted, you have reviewer comments, come consult with us then if there's room for expansion of your project. And we're not going to leave you hanging. So it's a collaboration. We embrace it. We're going to be there with you, watching as it goes through the various stages of a review, whether that's NIH, PCORI, Foundation, DOD, any. And we're excited when you get that answer of we're funded, or here's the score we reviewed, here's our pink sheets. We come back to consultation, let's digest it. How do we rehash it to then put it back in?
Very,
It's really a resource. It's really terrific.
it's
So-
very helpful about finding the blind spots.
One of the nice things about the NIH pathway and using the tin, and we didn't emphasize it enough. When you go look for those sites, we can go to all 60 major academic research centers, and the PI gets to do a presentation in front of the would-be site PIs. And that's very helpful because you get feedback of, sometimes it's you want to do what, and I don't want to do that, and you can't pay me enough money to do that. That's all helpful. So that's important. That's harder to do outside of the tin. But if we've done a trial in that area, we can go to the people we know.
Are there free educational resources, this being a nice support ground that we could, educate ourselves?
Trialinnovationnetwork.org is the website. There's a very large toolbox that is searchable that you can go and peruse and download.
You can download a bunch of templates and process diagrams.
Can you share that with Mary, please?
Yes,
Yeah.
of course.
Thank you.
Thank you very much.
Thank you.